Okay, you're good. Okay, so we're continuing on with evolution. We have our study sheet. We are good to go. Today what we're going to look at are, for some of you, this is going to be a little bit of a review with Hardy-Weinberg. We're not going to go into too much of doing work with the equation itself. We'll talk about it. But if you had honors bio, you guys did a lot of work with Hardy-Weinberg last year, or a couple of years ago. So we'll kind of go back over Hardy-Weinberg equilibrium theory and talking about that with microevolution, and then also the 5 assumptions, which I don't think we talk too much about in honors biology. So this is where we're going to get in a little bit more depth with Hardy-Weinberg. But the 5 assumptions of the Hardy-Weinberg equilibrium, and then the ways that we essentially break those rules, and how that affects populations, because we're still looking at how populations change over time. So the difference between these two pictures, what do you see? What's different about the picture on the left compared to the picture on the right? What do you see?
Genetic variation from the same
How is it varied?
feather color.
Feather color? Maddox, were you going to say something else?
Like one stayed the same and then one changed.
One stayed the same and one changed. Okay, good. Olivia, what were you going to say?
Well, I was to say one of the tails is longer.
One of the tails you notice is longer as one of the changes. Good. So we can see there are some noticeable differences between the two. Just looking at even between this change and this change here, we can see that, like you said, the tail is longer. There are some features that we notice that are not quite the same. So evolution, when we're looking at the definition of evolution, we're looking at changes in a population over time. And the big key thing that we want to take from that is that everything that's living today had to have come from some sort of ancestral species, and that those ancestral species are different from all of the present-day species. So with non-branching evolution, that was what we saw on the left. Maybe we had some differentiation with the color, but it's not a new species. Maybe it's just changing the color of the feathers. With branching evolution, though, this is where we could get new species out of this. We do, and that's where maybe because of these features here that are different, we start to see the new species come up. There are 2 types of evolution that we're going to look at, and we're going to split them between today and the next lecture. Today we're focusing on microevolution, and then the next time we're going to focus on macro. So microevolution. Those are the sweetest looking dogs. Microevolution, what are we talking about with microevolution? We're looking at changes in allele frequencies of a gene pool in a population over time. So the key thing here to remember is that the allele frequencies are changing. We have to remember that alleles are the different forms of a gene. So when we were talking about Punnett squares, and we had the big T and the little t, or a big G and a big G, the alleles are the different forms of the gene that you could inherit from your parents. And then gene pool, this word, we've heard this before, but technically, biologically speaking, we're looking at all the different types of alleles or forms of those genes that are in that population. And then the big thing to remember, this is going to be a big one for test time. Populations are evolving, not individuals. So when we talk about evolution and evolving, we're not looking at one individual changing over time. We're looking at this from a bigger scale of the whole population. big key thing that you kind of want to keep in mind here is that when we're looking at the ultimate source of having some sort of new genetic variation in a population, it ultimately is going to lead back to or lead to a mutation. So mutations, this is a really big concept to get from this. Mutations are any changes to your DNA sequence. We're literally changing the A's and the T's and the C's and the G's, which will then end up changing our mRNA strand, which then ends up changing our codons and the order that they come in to build that protein. And so when we have those mutations and we have something completely new, a new allele that comes from that, that's where a lot of this variation, where we have to remember this variation kind of, or will stem from all of that. Don't get that confused with crossing over. Crossing over, a lot of people think, oh, when the alleles or when the pieces of the chromosomes cross over in meiosis, it's not the same thing as having a mutation. Mutations, we're changing the A's, the T's, the C's, the G's, we're changing those orders of those bases, whereas with crossing over, it's just nothing's changed at that DNA level, at that molecular level. So not quite the same thing, and they won't lead to new alleles when we go through crossing over. Same alleles, just different way of getting it from a different parent or from one of your parents. So Hardy-Weinberg equilibrium, these are the two scientists who came up with this equation, really. But what we're looking at with the Hardy-Weinberg equilibrium theory is, and the equation, is basically our allele frequencies. When we're in Hardy-Weinberg, our allele frequencies aren't changing. And if we mess that up somehow, we violate that, we break those rules, then that's when we would see evolution occur, because those allele frequencies are going to change over a long period of time. So if a population is in Hardy-Weinberg equilibrium, allele frequencies are not changing at all for a specific trait, the population is not considered to be evolving because it's not changing over time. Mathematically, the baseline for a non-evolving population came from Hardy-Weinberg. They were the ones that proposed that if we're not changing, we're not evolving, then we are in this equilibrium here. And in their equilibrium, we have the variables P and Q. P is for one allele, Q is for the other. So if we have P, the number of P alleles and the number of Q alleles equaling, balancing out, then we're in equilibrium. The equation itself, so this would be for homozygous for one of the alleles. The 2pq would be heterozygous, so capital and a lowercase letter when we think of it in those terms. And then the q squared would be the homozygous allele for the other allele. Maybe it's the recessive one at that point. Again, this is strictly sticking to nothing is evolving in a population, nothing is changing with the allele frequencies. We're pretty much balanced between the P's and the Q's are balanced. Allele frequencies are not changing over time. If an allele frequency for a trait is not changing and if evolution is not occurring, then what do we think is true of this population? So what Hardy-Weinberg equilibrium tells us, this is our frequency of our dominant allele, the frequency of the recessive allele. They're in balance with each other. Nothing is going more one way or more the other way than the other. They're in fair equilibrium here. Then we should not see anything changing within the population. The allele frequency should stay the same time. But it could go, we could break those rules. So let's take a look at them first though. So the way that this works, if we follow these rules and everything is kind of met, we meet these standards, we should not see a population change over time. Those 5 rules or assumptions, we call them assumptions, are that we have to have a very large population. So we're looking at thousands and thousands as far as population goes, not a very small sample like 5. Or if we were to look at the school, for example, we would want to look at the entire school, not just everyone that's in this 3rd period class as a sample size. So we want to look at a very large population. There can't be any gene flow occurring. We're going to talk about all of these in a little bit here. We can't have any mutations occurring whatsoever, so everything needs to stay exactly the same with the DNA sequences on a molecular level. There needs to be random mating, it can't be selective, and there cannot be any natural selection occurring. If we meet all of those assumptions, then we will not see the population change over time. We will be in Hardy-Weinberg equilibrium. This gets to be interesting for a biologist in quite a few different ways, because once you do break one of those assumptions and we move away from them, then we'll start to see that allele frequency shift a little bit and change, and then we start to see evolution occur. Aha, here we go. So how do populations actually break one or more of those assumptions for Hardy-Weinberg? The first way that we can break one of those assumptions is through genetic drift. And genetic drift is our evolutionary mechanism where a population gene pool changes due to random chance. And by random, we mean this is totally random when things happen here. It could also be due to a sampling error, essentially is what we look at here, because we're not looking at this from a larger population perspective. It's kind of more of a smaller selected, but randomly selected gene pool or population set that we're looking at. I guess one way to look at it, think of it this way. If you were to flip a coin 1,000,000 times, out of those 1,000,000 times, you're probably going to have a good chance of pretty close to 50% of that was heads and 50% was tails, right? It'll be pretty close to that. But if you're flipping the coin only 5 times, you guys can get 5 coin flips in a row that are heads. Is that true for the probability of getting heads and tails then? No, I mean, you don't see a big enough selection in order to get that 50-50 chance or probability to show you. So what we're looking at in this first picture here with this generation is, let's say that we select out of all of these flowers, we select these 5 out of the 10 to reproduce with each other, to mate with each other and have offspring. In this case here, the frequency of having red flowers was 70%, and then the frequency of white was 30%. But if I only pick 5 and allow them to mate, I'm not getting a true representation of all of the types of flowers here. So then the results are going to kind of lean more towards maybe one of these. And so let's say that we pick those 5 and we let them mate, and then in the second generation, now we have closer to 50-50 red versus white. Okay, that's great, but now in the third generation, what I'm going to do is I'm only going to pick 2 from the second generation. Again, a sampling error perhaps, because I'm just randomly picking out 2 flowers and allowing them to mate. And when we do that, because of the 2 that I happen to pick, I have entirely red flowers now and no white. In nature, that wouldn't necessarily occur if everything was following the rules correctly, but because of this sampling error or this weird random chance, we end up with all red flowers instead of some white ones in there. So that's where we tend to see some issues. This usually gets caused either by a bottleneck effect or by the founder effect. I know we've kind of talked a little bit about both of these before in the past, so we'll kind of go over them again. But with the bottleneck effect, if we were to say that this bottle right here was a representation of all the different types of one population, something, usually it's like a virus or something that can maybe kind of cause this population to go towards extinction. But let's say that in this original population, we have green, we have orange, and we have purple organisms. But because of some event, maybe like a virus, very few of them are able to escape from that bottle and survive whatever that event is. And now if you look at which ones did survive, we have mostly purple, one green species, and none of the orange ones. And so that bottleneck effect causes quite a few, the orange went extinct, quite a few organisms to not be able to survive. The ones that do will be the ones that go on to reproduce. And because of that, now we've got a smaller sample of the green, absolutely no orange, and then that can cause things like genetic drift to occur. There's also the founder effect that can happen. So founder effect is when we have a population that kind of is living in an area, and maybe some of the population moves to a new area, and then they settle there and they live there. In this particular case here, in this little island, there were a small amount of people. I'm gonna check my notes. What did I say? Oh yeah. Roughly 12 people moved to this island, and a couple of different families, they moved to this island and then they colonized this island. This is their island now. But what happened was, at least one member of the family, one of the families that moved here, had retinitis pigmentosa. And retinitis pigmentosa causes you to lose your vision over time, but you don't know it until later on in life. And so it's not something that they really knew at the time when they went here, but because it's a small island, only a couple of families, there tends to be a lot of inbreeding between the families because it's on a small island and it's pretty remote. That means that a lot of the population is going to end up with this genetic disorder as well. And so the founder effect can cause certain traits to be more prevalent when it wouldn't be if we had a larger population to also to work with for reproduction. Okay, gene flow. Gene flow is another change that can cause our allele frequencies to shift and cause us to see the results, or to have some results in evolution. With the gene flow, populations can gain or lose individuals that are fertile, meaning that they can reproduce. Due to some sort of movement of the population in or out. So immigration, we're having our population move into a new area. Emigration, the population will move out of the area. So when we have these movements of these fertile individuals leaving or coming into an area, that definitely changes which genetics now are part of the gene flow, which ones are coming into play with all of that. Oh, whoa, back up. Are we good here? No, not yet. If we have part of our population moving out, they take with them those genes. If they were genes that were more favorable and more selected, then we lose access to that as far as increasing our numbers in population during reproduction. And then moving in, that can change too, because if there is a trait that is, or an allele that is more favorable that wasn't, and now this is what's to choose from for reproduction, in higher numbers of that, then we can see an increase in those alleles as well. All right. Mutations can change gene pools. Again, this is where we're changing the organism's DNA. This is at that molecular level. Where the A's and the T's and the C's and the G's are going to change. this could be due to a lot of different reasons, things like UV light. We see this with humans a lot, in pigment and skin cancer that can change things one way or the other. Different mutagens can also affect that. Mutations are a big one that can cause change in the gene pools, shifts in the gene pools. And then the fourth one here, this one can be due to either what we call non-random mating or sexual selection. This is one that definitely violates Hardy-Weinberg, because Hardy-Weinberg says that there has to be random mating. This would be not random. This is where maybe one particular individual is chosen as a mate more than any other, or more than just others, and then that one organism keeps having, that one individual keeps having offspring. So if there is a male that is chosen to have more babies with more females, then his traits are going to be obviously higher in number than the other males that are there to choose from. Again, it's not random, so this gets to be a little bit of a problem, and this will definitely get us out of Hardy-Weinberg equilibrium because one set of traits is favored over others. Yeah.
Do we see this in like a lot of cattle and stuff? With
A lot of what?
cattle? Or like
Cattle?
like cows and everything? There's like one bull.
That would be breeding, like selective breeding, I think. Is that what you're thinking,
Yeah.
more or less? Yeah, I mean, when we're doing selective breeding, we're definitely not in Hardy-Weinberg equilibrium, because those traits are being favored. And so like if you're studying out a dog or a cow or whatever, yeah, it definitely will not be in equilibrium, because those traits are going to keep getting passed on, and they're going to be out there in more multitudes and higher population with the offspring. Yeah, definitely. The last one, I think this is the last one. Natural selection. So to be in Hardy-Weinberg equilibrium, no natural selection has to be occurring, but a lot of times natural selection is occurring, and so that's where we can start to see those changes with our allele frequencies. We have a few different types of selection that we typically tend to see in biology. There's directional selection. This is extreme forms of a selection. So one way to think about this example we have is if you have a giant field, and this field is full of mice, and we have black mice, and we have white mice, and we have gray mice, directional selection means that we would pick one of those extremes. So either the black mice or the white mice would be the ones that are that seem to be mating and reproducing and having more offspring. So it would be one of the extremes, either black or white. Disruptive is where it would pick both of those colors, the black and the white, and they would be mating and they would be reproducing, and we would have lots of black mice and lots of white mice, but very few gray mice. And then the last one is stabilizing selection, and this is where we finally pick that intermediate form, and the gray mice population is now increasing over the black or the white mice. And remember, when it comes to natural selection, the whole picture and piece of natural selection isn't just survival of the fittest, it's surviving and then reproducing to continue to pass on your genetic traits to your next generations. And so when we have any of these types of selection, where we have one form or another being picked for reproduction, and it's more extreme that way, then that really shifts things shifts those alleles, those frequencies, to favor one over the other. All right, so why is this really important and interesting for biologists? Well, it really helps us to look at evolution and understand how evolution occurs, and kind of helps us to see, okay, here's where this is going, and this is where it could be going, and where it's going to change here. When you're in Hardy-Weinberg equilibrium, the population is not evolving. It's not changing at all. The allele frequencies are pretty much staying the same. You have equal amounts of dominant allele or recessive allele and heterozygous, and everything is basically in balance. But again, you have to have all of these met. So large population, no gene flow, no mutations, random mating has to be happening, and no natural selection can be occurring. To have all of those assumptions met is really tricky though, because a lot of times you're going to see that this is not easy to accomplish. And so if you break even one of the Hardy-Weinberg assumptions, now we start to see some changes, and we can start to see some evolution occurring. Whoa, back up. Hold on a minute here. There we go. Small populations can lead to genetic drift, or can cause genetic drift through things like either that founder effect, or through, what was the other one that we talked about? The founder effect, or the bottleneck effect. We have gene flow. Mutations are constantly occurring, and when we have those constant occurrences of those mutations, if they're getting passed on, that's definitely going to change things. A lot of mating goes on where there is selective mating, and it's not as random as it could be, and then also natural selection is constantly occurring as well. There was something I was going to talk about. I wonder if it was in here maybe. Oh yeah, it was here. So going back to this really quick, because I just popped back into my head. Did you guys learn about the peppered moths last year? So, okay, if you had my class, we talked about peppered moths. I don't think Mrs. Lewis brought it up much, because, I don't know, for whatever reason. When we have natural selection occurring, or is something like that, it's usually because of, or any of these three types of selection, it's usually because of some sort of a pressure was put on the organisms to change and be kind of forced to pick one of these extremes, or to pick the intermediate. With the peppered moths, so this is a good one to kind of go over with you guys. During the Industrial Revolution in Britain, they had all these moths that were kind of a white with black speckled colors on them, and they essentially looked like the lichen that would grow on the trees there, because there were a lot of trees that had a lot of lichen growing on them. So these moths would sit on these trees, and they would blend in with the trees because they looked like the lichen, and predators would not pick to eat them. But then during the Industrial Revolution, so that was pre-revolution, during the Industrial Revolution, there was a lot of soot coming from when they were burning a bunch of coal. And all of that soot goes up into the air, causes a bunch of pollution, it lands on the trees, and now the trees, or the lichen, I should say, stop growing on the trees. They're not going to be growing there. The trees are starting to become more darker in color. And now these peppered moths that are sitting on these trees are standing out on this black background. You have this white moth, and now predators see them, and now they want to go and eat them. So they can't. They go pick them out really easily. And so over the years, we saw this shift happen due to natural selection, where those moths that were darker are now able to survive longer. They can get chosen as mates, and they can pass on their genetic information to their offspring. And this kept happening over a long period of time. And then eventually, we realized, oh, this is not good for the environment. And so laws were passed. And then once those laws were passed, less pollution, less black soot on the trees, and now you have black moths on lichen covered trees again. So what's gonna happen? Huh?
Predators are gonna eat them.
Yeah, they're gonna get eaten. And now which trait is more favorable to have? The peppered moth, white ones. And so they're gonna be the ones that are gonna get selected. So we're looking at these extremes here, and that's just some sort of environmental change. You see that a lot, populations have to do this. A lot of times there's a lot of human influence involved, but a lot of populations have to change and maybe pick one form over another for an allele in order to survive because of human interaction. That could be stuff like climate change. Around here, we see this a lot with our populations when it comes to hunting and fishing, based on things that we do that affect their environment. So all of that stuff kind of leads back to this with the natural selection, and that's where it gets really hard to not be evolving. Is kind of difficult to do. We're constantly having to deal with this natural selection piece. And when we can break Hardy-Weinberg, we do get evolution. Changing over time in populations, not individuals. Okay, we're matching now. You guys ready to match? Did everybody get your boxes filled in? Are we good? Okay. I'll post these today so that way if you miss something you can go back and look. But I can go back if I need to go back. Are we good? No? Alright, I think some of you have already started filling these in. Number 1, differential survival and reproduction of certain individuals over others in a population. What do we call that?
Natural selection.
Natural selection, very good. Number 2, the ultimate source of new genetic variation in a population. Mutations. The ultimate source of this is always going to go back to those mutations. Good. Number 3, type of selection that favors selecting an intermediate form within a population.
Stabilizing selection.
Stabilizing selection, very good. Number 4, movement of fertile individuals into or out of a population changes the gene pool. Gene flow, good. Immigration, emigration. And evolution, number 5. An evolutionary mechanism where the gene pool changes due to random chance. Good. Genetic drift. And then number 6, a form of natural selection where certain individuals are more likely to mate than others. Good. G for that one. Number 7, type of selection that favors selecting opposite extremes over an intermediate form.
Disruptive selection.
You guys are on a roll. Number 8, type of genetic drift caused by a near extinction event in a population. Good. And then number 9, type of selection that favors selecting one extreme form within a population. Good, directional. And then 10, type of genetic drift caused by the establishment of a new small population away from the parent population.
Founder effect.
Founder effect, great. Do you guys have any questions? We're good? We're good? We're done? That's it?